I'm Kate Rochlin, the Chief Operating Officer at IN8BIO, and today I'm going to tell you the story of how we've brought gamma delta T cells from observation into clinical therapeutic. Ooh. Or not. There we go. So lawyerly disclaimers. So unfortunately, our story starts like so many stories in oncology, and it starts with the story of a child that we couldn't save. And so this little trinket here that you see is actually a Christmas wreath that was made by a 6-year-old patient for our Chief Scientific Officer, Dr. Larry Lamb, 30 years ago. And this led him on a journey to understand why some of his haplotransplant patients lived and why some did not when they seemed similar on paper. And that led him to the gamma delta T cell. So I'm going to tell you about how we've gone from that observation to what we've been able to bring into the clinic today. And we truly believe that using the gamma delta T cells synergistically with other therapies, we can drive towards cancer zero, which is our mission of eradicating every single cancer cell from a patient in an ideal world. And we believe there are 3 pillars that you really need to be able to have an effective cell therapy. The first is durability. We need these treatments to persist, and we know that that's been an issue in many of the innate cell therapies. So I'm going to talk to you today about how we believe we can achieve that durability. Tolerability has been an issue in cell therapy. How do we develop something that's effective but also safe and well tolerated in the patient? And finally, heterogeneity. How do we tackle tumor heterogeneity, particularly in the solid tumor space, where many, many very smart people have spent years looking for targets that are expressed solely on the tumor and not on healthy tissue. And we at IN8BIO believe that maybe those targets don't exist. So how do we tackle those heterogeneous, complex tumor microenvironments? And that's brought us to the gamma delta T cell. The gamma delta T cell is actually a really unique immune cell. It sits at the nexus of the innate and adaptive immune system, and as such, it actually has properties of both. It can kill very similar to an NK cell through TRAIL and FAS ligand, but it can also antigen present and drive deeper immune responses similar to the adaptive immune system. Importantly, it is not MHC restricted, so it can go from donor to a recipient with no modifications, and importantly, its natural function is actually to differentiate between healthy and sick or healthy and stressed tissue, such as tumor tissue. And it does this through the NKG2 ligands, NKG2D receptors, as well as the TCR. And those combinatorial receptors allow it to very exquisitely detect healthy versus not healthy tissue. Gamma delta T cells have also been shown for many years to be correlated with improved survival outcomes. So what I'm showing you all the way over there on the right is actually 20,000 patient analysis showing things that were associated with favorable versus unfavorable outcomes. Gamma delta T cells are right at the top. That graph I'm showing you there in the middle is actually our CSO, Dr. Lamb's study that came out of his observations with that 6-year-old patient, showing that patients with high levels of gamma delta T cells had improved survival over those with low gamma delta T cells. And then finally, I'm showing you that that also holds in solid tumor settings. And here I'm showing you a human example of colorectal cancer, where you see high gamma delta T cells correlating with survival. So how do we take something that we've seen observationally and turn it into a therapeutic? And that's what we've done with our Deltex platform. So our Deltex platform is all ex vivo expanded activated gamma delta T cells. And there are a couple of unique components to this platform. The first is our ability to expand gamma delta T cells at scale. We can do both V delta 1s and V delta 2s, but the clinical programs I'm going to tell you about today are primarily V delta 2. We've also developed first-in-class genetic engineering, and I'm going to tell you about 2 approaches today. One is a more traditional CAR approach, which will be familiar, with a little twist. And the other is actually a way that we've engineered our cells to be chemotherapy resistant. And we call this drug-resistant immunotherapy. We actually hijacked a tumor's own resistance mechanism, put that into a gamma delta T cell, which makes it chemo-resistant, so we can dose it with chemotherapy, where chemotherapy will drive stress, and that lights up those cells like a Christmas tree for our gamma delta T cells to seek out and kill them. And then finally, we've brought it together in a closed manufacturing system that's operating at clinical scale. Here's our pipeline as it stands today. We have our INB-200 program, which I'll tell you about. These are our chemo-resistant cells that we're using in a phase 1 trial for glioblastoma. We have INB-100, which is an allogeneic, non-genetically modified cell that I'll tell you about our work today in leukemia patients. INB-400 will be the phase 2 portion of INB-200. That was just cleared by the FDA last December. And then INB-300 is our non-signaling CAR, which I'll touch on very briefly. We also have an IPS program, which I'll not be talking about today, but happy to answer any questions later. So going back to the beginning, how do we overcome this really difficult problem with solid tumors? How do we deal with this really inhospitable tumor microenvironment with little T-cell trafficking? And we are going to tell you about INB-200 today. So we've gone into glioblastoma, and we get a lot of questions about why glioblastoma. And actually, the biology and the mechanism make a lot of sense here. Glioblastoma is known to be very upregulating with stress signals. So we know that it's very identifiable to gamma delta T cells. It also occurs in the brain, which is considered an immune-privileged environment. So we can actually take our gamma delta T cells and infuse them directly into the tumor bed, so we know that they're actually getting to the right place, which is critical in solid tumors and tumor homing. And finally, the standard of care in glioblastoma is TMZ. So TMZ is actually lymphodepleting. So we don't have to use a conditioning regimen. We can go right into the frontline setting and use our gamma delta T cells with the standard of care. TMZ also functions to upregulate the stress signals on those tumor cells even more. And here I'm showing you a little bit of that data, that's our preclinical data. But what you can see is in even chemotherapy-resistant glioblastoma cells, you can see that the addition of TMZ massively upregulates multiple stress ligands. So again, helping us tackle that tumor heterogeneity. Because we don't have to recognize one target, we can recognize 8 to 10 stress signals. And then I'm also showing you that in the stem-like cells in that population, we can also upregulate stress. So we can target chemosensitive, chemo-resistant, and stem-like cells. So this is what our trial looks like. It's a fixed dose frequency escalation trial. So cohort 1 gets a single dose of gamma delta T cells, cohort 2 gets 3 doses spaced 28 days apart, and cohort 3 gets 6 doses spaced 28 days apart. And so we're looking at safety and MTD here. And so this is quite a busy slide, and unfortunately I'm showing you a data cutoff from last December. We are actually going to be updating on this trial at ASCO, so stay tuned. But even in this limited data cutoff that I'm able to show you here, what I hope you can see in this slide is that every patient that we've treated has actually gone beyond their expected progression-free survival, with many patients going beyond their expected overall survival. And I will note that unfortunately, patients 1, 7, and 14 all passed away from something that was not their glioblastoma. So none of them died of progression of disease. So we're continuing to enroll, and we'll be updating this at ASCO this year. But what I want to show you is actually a really interesting data point here on persistence and how our cells may be working. So we had one patient that was re-resected. So they had their initial resection, and that's represented in A here, and we're showing you a negative image in the black and white. And it's showing you that there are essentially no gamma delta T cells in that space, that this is, again, a very immune-privileged environment. It's very resistant to T cell trafficking. You just don't tend to get the cells going into a glioblastoma. In B, that's the actual re-resected tumor from our patient. And you can see that even over 100 days beyond our infusion of a single dose of gamma delta T cells, there's significant infiltration in that tumor, and actually more than 50% of what the surgeon removed was dead necrotic tissue. So even with a single dose, it appears that 100 days later, we're seeing significant gamma delta T cells infiltrating and functioning in that space. So INB-400, I'm simply going to say that this is our phase 2 trial based on this technology. It was cleared last December, and we're currently getting Arm A up and running, which is in newly diagnosed glioblastoma patients, and that should be enrolling later this year. I'm going to switch gears and go back to the study INB-100 that actually came directly from that observation from that 6-year-old girl who did not survive. So this is a study using non-modified allogeneic gamma delta T cells in patients who are undergoing haploidentical stem cell transplant. And in this study, we're taking exactly the observation that Dr. Lamb made and using it therapeutically. So this is a single dose escalation study. So 1 times 10 to the 6th, 3 times 10 to the 6th, and 1 times 10 to the 7th. And these patients are receiving their transplant, and then they're receiving an extra dose of gamma delta T cells right after engraftment to seek out and eliminate any residual leukemic cells. And we actually know that there are leukemic cells in many of these patients because haplotransplants get a reduced conditioning regimen. And that leads to high rates of relapse. So you're trying to balance the graft versus host and the host versus graft. So you get about 50% relapse at 1 year, and in some populations up to 85% relapse at 2 years. Again, I'm showing you a data cut from last December, and this will be updated at EBMT in about 2 weeks. And what we are thrilled about is all the patients here to date are still in complete remission following our treatment with gamma delta T cells. So you can see we have patients out beyond 2 years, 1 patient approaching about 1.5 years, and several here that were just dosed. So this is extremely encouraging to us because these players were not good transplant candidates. Many of them had failed multiple lines of treatment, multiple transplants, and CAR Ts prior to joining our study. So this data will be updated next week, and not only will we demonstrate long-term remissions, but we'll also give some data on durability and gamma delta T cell proliferation post-transplant. So stay tuned for that. And in my last couple minutes, I'm going to switch gears yet again and tell you about INB-300. And so how do we take gamma delta T cells and use a CAR? Because what we don't want to do is put a CAR on a gamma delta T cell that limits it. And what I mean by limits it is at the beginning I told you that gamma delta T cells had multiple receptors that could recognize stress. And through that combinatorial recognition, it could very sensitively detect healthy versus sick tissue. And if you put a CAR on that targets it to only a single antigen, you can abrogate that whole system, where then it can only recognize and kill based on that single target. So what we do instead is actually a non-signaling CAR approach. So we essentially have designed a CAR that goes on the gamma delta T cell, but has no intracellular signaling domain. So the CAR itself does not induce the gamma delta T cell to kill. It solely acts as a GPS to bring the gamma delta T cell in close contact with the tumor cell, and it will only then kill if it also engages the stress ligands on the surface of that tumor cell. So it's essentially a 2-part system in a single cell. And what that allows us to do is when the gamma delta T cell binds an on-target but off tumor ligand, it should not kill because that healthy cell will not be expressing those stress signals the way that a cancer cell would. And so this is just a schematic of how that works here. And I'm just going to show you a tiny bit of data that's from a proof of concept in vitro study that we did using CD19, which is a verified target. We're not actually taking CD19 forward in the clinic. We're actually releasing data on our proposed targets and additional data on this program at AACR next week. So unfortunately, all I can give you is CD19, but we have some very exciting data coming out on this program very shortly. And here, what we're showing you is that using a CD19 non-signaling CAR, that we can actually discriminate between a NALM6 CD19 expressing leukemia line and healthy B cells, which also express CD19. So we can actually cause killing of the tumor cell line, but spare the B cell line, demonstrating that using this non-signaling CAR, we can actually differentiate between these 2 cell types. And we're really excited about this approach because what this allows us to do is to, 1, potentially rescue targets that companies have identified that were really good targets for targeting tumors, but had on-target off-tumor effects that led them to be toxic. It also allows us to treat what were previously considered undruggable targets on tumor cells, where we knew that there was low-level expression on healthy tissue. So we're really excited about the potential of moving this platform forward in solid tumor and liquid tumor indications. And again, we'll be releasing some data on that at AACR next week. And with that, I just want to thank you so much for hearing the summary of our programs, our work in gamma delta T cells, and how from an observation 30 years ago, we're very, very proud to be working with Dr. Lamb to advance these programs in the clinic and continue to help patients on our mission towards Cancer Zero. Thank you so much.